Welcome to the Deep Dive. This is where we take your sources, could be articles, research papers, maybe even just notes, and we really pull out the key insights for you. Today, we're jumping into the microscopic world, an organism that, well, it really shows that complicated, sometimes contradictory nature of our own microbiome. We're talking about Bacteroides stercoris. Now, this is maybe an organism that flies under the radar a bit sometimes, but clinically it's actually really significant, especially because it's a key member of that, notorious Bacteroides fragilis group. So our mission today, to make sure you, our medical microbiologist listeners, are really up to speed on this crucial anaerobic bacterium. This whole deep dive is powered by a Gemini deep search, and if you happen to be watching those amazing visuals, they're courtesy of Microbiome. And like always, all the details, the primary sources, the citations are all laid out for you in the notebook document that comes with this. Okay, let's unpack this thing.
Yeah, let's do it. So Bacteroides stercoris. Fundamentally, what are we looking at? It's a gram-negative rod, an obligate anaerobe, meaning no oxygen, and it doesn't form spores. And it really is this perfect example of that dual nature you mentioned, kind of a Jekyll and Hyde situation.
How so?
Well, on one hand, it's massively abundant in our colon, huge numbers, and it's metabolically super active, really beneficial for gut health, a true commensal.
Yeah.
But if it gets out of the gut, if those barriers break down, then it switches roles. It becomes an opportunistic pathogen, and it can cause some pretty serious infections.
And being part of the Bacteroides fragilis group, that name alone sets off some alarms in microbiology, doesn't it? Why is that group such a big deal?
It really does. The B fragilis group is, well, notorious is the right word, because collectively these are the anaerobes we isolate most often from clinical infections, period. They get that reputation because, one, they have significant virulence factors, ways to cause disease, and two, and this is huge, their antimicrobial resistance is widespread and constantly evolving. It's a major challenge.
So B stercoris inherits that family reputation.
Exactly. And you often find B stercoris involved in polymicrobial infections, meaning lots of different bugs working together, especially in complicated intra-abdominal infections. CIAI is the shorthand. Think peritonitis, abscesses deep in the abdomen. These usually happen after something disrupts the gut integrity, like, abdominal surgery or trauma, maybe appendicitis, diverticulitis. Something lets it escape.
So it's rarely the solo villain, more like part of a gang. What makes it such a tough member of that game to deal with clinically?
Right. It thrives in that mixed environment. Yeah. The challenge comes from, well, two main things. First, it's got some effective virulence factors, things that help it survive and cause damage. And second, its antimicrobial resistance profile. It's extensive and it keeps changing.
Let's break those down. Virulence factors first.
Okay, so a big one is its polysaccharide capsule. Think of it like a protective shield. It helps the bacterium evade phagocytosis, getting eaten by our immune cells. That's critical for it to establish itself. Plus, it has ways to mess with our immune response. It can modulate its surface to avoid recognition, and it can even decrease things like nitric oxide production in macrophages, which normally help kill bacteria.
Okay, so it's slippery, defensively strong, and the resistance side.
That's where it gets really complicated. It has intrinsic resistance, meaning it's naturally resistant to certain antibiotics just because of its basic biology. Yeah. But it's also incredibly good at acquiring new resistance mechanisms. So we see built-in resistance to many beta-lactams, but then on top of that, we're seeing more and more acquired resistance to drugs like clindamycin. And even though thankfully less common for stercoris compared to fragilis itself, we're seeing resistance pop up to carbapenems and metronidazole. That's really worrying.
Wow. A common gut bug acting as a reservoir for resistance. That feels like a major public health issue waiting to happen.
It absolutely is. And that's maybe the most critical insight for us as medical microbiologists. Because it lives in the gut, this incredibly dense microbial environment, B stercoris is perfectly placed to be a hub for horizontal gene transfer. These resistance genes, they're often on mobile genetic elements, plasmids, transposons, things that can easily jump between bacteria.
So it can pick up resistance from its neighbors and potentially pass it on too?
Precisely. It can acquire resistance and it can potentially disseminate it to other gut bacteria, maybe even more traditionally nasty pathogens. Yeah. So when we track B stercoris resistance, we're not just thinking about treating that one infection. We're actually monitoring a sentinel, an early warning system for emerging resistance threats that could impact a much wider range of infections.
That completely reframes its importance. It's not just another isolate, it's an indicator. So if we do have an infection involving B stercoris, what are the absolute must-dos for management?
Okay, the number one non-negotiable principle is surgical source control. You have to deal with the source of the infection. Drain the abscess, remove the dead tissue, fix the leak in the gut, whatever it is. Antibiotics are critical, yes, but they are adjuncts to source control. They can't sterilize a big collection of pus or dead tissue on their own. And the antibiotic therapy itself, it has to be guided by guidelines, informed by likely resistance patterns. Remember that intrinsic resistance, and crucially, you have to assume it's a polymicrobial infection. You need broad coverage. Now, to really get why it behaves the way it does, understanding its taxonomy, its place in the bacterial family tree is really helpful. It gives us clues about its inherent traits. It was first properly described back in 1986, and the species name, Stercoris, it literally means of dung or of feces.
Huh. Doesn't leave much to the imagination, does it? Tells you exactly where it lives.
Pretty much. It's very descriptive.
So walk us through its official address, kingdom, phylum, all that jazz.
Okay, starting broad. It's in the domain Bacteria. Then phylum Bacteroidota, class Bacteroidea, order Bacteroideales, family Bacteroidea, genus Bacteroides, and finally species Bacteroides stercoris. And the official reference strain, the sort of type specimen for the species, is ATCC 43183. That one was originally isolated from a human fecal sample, naturally.
Okay, we've got the basics. Now, here's where it gets really interesting, I think. Getting that genetic blueprint, its taxonomy, to how it actually acts in a clinical setting. How does the genome translate to behavior?
That's the key connection. So genomically, Bacteroid species generally have a G plus C content. It's the proportion of guanine and cytosine in their DNA, somewhere around 40 to 48%. For B. stercoris specifically, if we look at the sequence genome of strain BFG121, it has a single circular chromosome, about 3.88 million base pairs long. And on that chromosome, it encodes something like 3,081 protein genes and 83 RNA genes. That's a pretty hefty genetic toolkit.
And that toolkit allows it to do what exactly?
It underpins its metabolic versatility. It lets it break down a huge range of complex molecules, survive in different environments from the comfy gut to the much harsher conditions inside an abscess. It's adaptable. But, and this is crucial for you in the clinical lab, the single most important piece of taxonomic information isn't the exact genome size, it's its membership in the Bacteroides fragilis group.
Right, you mentioned that earlier. Why is that group designation so practically important?
Because it's not just a label. It's like a diagnostic and therapeutic shorthand. As soon as you know, or even strongly suspect, an isolate belongs to the B. fragilis group, you instantly know some key things about it. For instance, you know it's almost certainly resistant to 20% bile. That's why BBE agar works. And you know it also certainly produces a chromosomal class A cephalosporinus.
The CYP gene one.
That's the one, encoded by CYPA. And that enzyme reliably knocks out penicillins and many of the earlier cephalosporins. It's intrinsic resistance you can count on being there.
Okay, so just knowing B. fragilis group tells the clinician, don't bother with penicillin G or maybe cefazolin alone.
Exactly. It immediately guides empiric therapy before you even have the final species ID or full susceptibilities back. It points towards using a beta-lactam beta-lactamase inhibitor combo or a carbapenem or metronidazole drugs that bypass that CPA resistance.
That's a powerful shortcut in a critical situation. Turns taxonomy into immediate clinical action.
Precisely. It's applied taxonomy, making a real difference at the bedside.
So what does this all mean for us as medical microbiologists when we think about how it causes disease? Let's dig into those dual roles again. The good side first, its job as a commensal.
Right. Back in its home turf, the colon, B. stercoris, is a model citizen, a vital mutualist. It's incredibly abundant, like we said, easily 10 to the 10th, even 10 to the 11th, bacterial cells per gram of feces, a major player, its main contribution. Metabolism, especially carbohydrate metabolism. B. stercoris, like its Bacteroides cousins, is packed with enzymes called Czymes carbohydrate active enzymes.
None of those let it break down.
Complex plant polysaccharides, dietary fibers, stuff our own human enzymes can't touch. It ferments these complex carbs.
And the payoff for us? The host.
The main payoff is the production of short-chain fatty acids, or SCFAs. Think acetate, propionate, and especially butyrate. These SCFAs are fantastic for us. They get absorbed and used as energy directly by our colon cells. They contribute to our overall energy balance systemically. They have anti-inflammatory effects, and they play a big role in modulating our immune system.
So a gut full of things like B. stercoris is generally a healthy gut.
Generally, yes. A high abundance of bacteroidates relative to another group, the firmicutes, is often linked to being leaner and having better metabolic health. There's even research specifically linking B. stercoris to better glucose sensitivity, and people have explored it as a potential probiotic for obesity. So yeah, definitely beneficial in its proper place.
Okay, paints a nice picture. But we know there's a dark side. When does this beneficial friend turn into a dangerous foe? What's the trigger?
The trigger is always a breach of the mucosal barrier. It has to escape the gut. And importantly, these are almost always endogenous infections. The bug comes from the patient's own gut flora.
So not usually caught from someone else.
Very rarely, if ever. It happens when there's damage, abdominal surgery, like we mentioned, penetrating trauma, or diseases that perforate or weaken the gut wall, like appendicitis, diverticulitis, maybe inflammatory bowel disease, or even cancer. Once it gets into a normally sterile place, like the peritoneal cavity or the bloodstream, then it deploys its virulence factors to survive and set up shop.
Let's revisit those virulence factors. What exactly helps it establish infection once it's escaped?
Okay, key factors. We mentioned the polysaccharide capsule that's huge for avoiding phagocytosis, surviving that initial immune onslaught. Then there's immune evasion. It can change its surface molecules to be less recognizable. It can dampen the killing mechanisms of immune cells, like reducing nitric oxide and macrophages. And another interesting one is its aerotolerance. Even though it's an obligate anaerobe, it can actually tolerate oxygen for a short period.
That seems counterintuitive for an obligate anaerobe. Why is that important?
It's crucial for that initial phase. When the gut perforates, the peritoneal cavity, for instance, still has some oxygen. That brief aerotolerance allows B. stercoris to survive those first few minutes or hours. Long enough for other bacteria, the facultative anaerobes, to use up that oxygen and create the truly anaerobic environment. B. stercoris needs to really thrive and multiply.
Ah, that brings us back to the polymicrobial nature. It rarely works alone, you said.
Almost never. It's the ultimate team player in infections. You'll almost always find it alongside facultative anaerobes, think E. coli, Klebsiella, maybe Enterococcus, and often other strict anaerobes too. I mean, this isn't random, it's pathogenic synergy. The facultative bugs do B. stercoris a huge favor by eating up all the oxygen, lowering the redox potential.
Creating the perfect anaerobic conditions.
Exactly. And then everyone contributes their own nasty tools to the fight enzymes that break down tissues, toxins, ways to mess with the immune system. It's a combined assault. And that synergy is precisely why your empiric antibiotics have to cover both the facultative guys and the anaerobes like B. stercoris. You need to hit the whole team.
That teamwork aspect makes treatment inherently more complex. So clinically, what specific diseases are we talking about where B. stercoris is a frequent player in this team?
Given its origin, the diseases are mostly related to the abdomen and pelvis. It's a major player in complicated intra-abdominal infections, so diffuse peritonitis or forming localized abscesses in the abdomen, liver, or pelvis. You culture an abdominal abscess, you'll often find B. stercoris in the mix. It's also common in surgical site infections, especially after GI or pelvic surgery. Makes sense, right? Potential contamination during the procedure. If things get really bad, it can lead to bacteremia and sepsis getting into the bloodstream. That usually happens when an intra-abdominal infection isn't contained. Less commonly, you might see it in bone infections, maybe diabetic foot infections, other soft tissue infections, often as part of that mixed flora.
And you mentioned a link to chronic conditions too. That's intriguing.
Yeah, this is a really interesting evolving area. Studies looking at gut dysbiosis imbalances in the microbiome have found lower levels of B. stercoris in people with inflammatory bowel disease, IBD. And in ulcerative colitis specifically, its abundance seems to correlate inversely with how active the disease is. Lower B. stercoris, more active disease.
So maybe its presence is protective somehow.
It suggests that, yeah. Or maybe it's just a marker of a healthier gut state. We don't fully understand the mechanism yet, but it hints at a role in gut immune balance that goes beyond just simple digestion. Still lots to learn there. Okay, let's shift gears to the lab. Diagnostics. And for anaerobes like B. stercoris, step one, maybe the most critical step, is specimen collection and transport. You absolutely have to get this right.
Because of the oxygen issue.
Exactly. Oxygen is toxic to these guys. So the best specimens are things like tissue biopsies or fluid aspirated directly from an abscess or a normally sterile site. Use a needle and syringe, you get the fluid out, and then immediately you need to get that sample into a dedicated anaerobic transport medium or vial, something designed to keep the oxygen out. Time is critical.
So standard swabs, probably not the best idea.
Generally, no. Strongly discouraged, really. Swabs collect a small volume, they dry out easily, and most importantly, they expose the bacteria to lots of air during collection and transport. You'll kill the anaerobes before they even reach the lab. False negatives are a real risk.
Okay, so good sample aspirate or tissue into anaerobic transport ASAP.
Yeah.
Then in the lab, first look is usually the gram stain, right? What are we looking for?
Yep, a direct gram stain from the specimen can give you quick clues. B. stercoris typically shows up as gram-negative rods, or maybe cocobacilli. They're often pleomorphic, meaning variable in shape. A key feature is that they often stain quite faintly, pale pink, and they don't form spores. You might see some vacuoles inside the cells sometimes. That pale staining and pleomorphism can actually be a hint that you might be dealing with anaerobes.
Right, so the faint staining itself is a clue. Now, getting it to grow. Given it hates oxygen, what are the specific conditions and media needed in the lab?
It absolutely requires a strict anaerobic atmosphere. That usually means an incubator or an anaerobic jar filled with a gas mix, typically nitrogen, some hydrogen, some CO2, to exclude all the oxygen. Temperature is standard body temp, 35 to 37 degrees Celsius. And under those conditions, you should see visible colonies, usually within about 24 to 48 hours, maybe a bit longer sometimes.
And what do the colonies look like on a standard plate, say, Brucella blood agar?
On a good, non-selective enriched medium, like Brucella blood agar, or maybe Columbia blood agar with sheep blood, the colonies are usually, not huge, maybe 1 to 3 millimeters across. They tend to be circular, convex, like a little dome with smooth edges.
Yeah.
And the color is typically grayish-white. Pretty standard bacterial colony look, really.
But then there's that special agar, the one that's really key for this group, BBE, right?
Yes, Bacteroides bilesculin agar. BBE is fundamental for presumptively identifying the B. fragilis group, including B. stercoris. It's a selective and differential medium, which is really clever.
How does it work? What makes it selective and differential?
Okay, selectivity comes from two things. It contains 20% bile salts. That high concentration inhibits a lot of other bacteria, including many other anaerobes, but the B. fragilis group is resistant. It also usually has gentamicin, which knocks out most facultative bacteria. The differential part comes from esculin. The medium contains esculin, and members of the B. fragilis group like B. stercoris can hydrolyze esculin, break it down. When they hydrolyze esculin in the presence of ferric citrate, which is also in the agar, it produces a black precipitate.
So that's where the characteristic look comes from.
Exactly. You get growth, because it's bioresistant, and the colonies themselves turn gray to black, and they're surrounded by this dark brown or black zone in the agar where the precipitate forms. Seeing colonies 1 millimeter that are black and blackening the BBE agar is a very strong, rapid, presumptive ID for the B. fragilis group. Huge help in the lab. Now, while those traditional methods like gram stain and BBE are still vital, modern identification has really moved forward. The absolute gold standard now for identifying a bacterial colony quickly and accurately, that's MEL-ADI, a mass spectrometry.
Right, MALDI-TOF. How does that change things?
It's a game changer. You smear a bit of colony onto a target plate, fire a laser at it, and analyze the proteins that fly off. It generates a unique protein fingerprint for the organism. Within minutes, you can get a reliable species-level ID. It's fast, accurate, cost-effective, really revolutionized routine bacterial identification.
But sometimes you need more, right, for tricky cases or research?
Absolutely. For definitive identification, especially if MALDI-TOF gets an uncertain result, or for taxonomic studies, or maybe identifying a really unusual strain, then 16S RNA gene sequencing is still the ultimate reference method. It gives you the precise genetic sequence for comparison.
Even with MALDI-TOF being so powerful, those classic biochemical tests and phenotypic characteristics still tell us a lot, don't they?
Yeah.
Especially for confirming group characteristics or differentiating within the group.
They definitely do. They provide valuable corroborating information. So let's recap the key lab characteristics for B. stercoris from that perspective. Gram stain. Gram-negative rod coccobacillus, often pleomorphic, often pale staining, oxygen, obligate anaerobe. BBE agar. Growth. Black colonies. Blackening the medium confirms bioresistance and esculin hydrolysis.
And those special antibiotic discs, the potency discs.
Crucial. These test for intrinsic resistance patterns. Classic for the group. You expect resistance, meaning no zone of inhibition around kanamycin, Bascom gram disc, vancomycin 5-gram disc, and colistin 10-gram disc. Seeing the RRR pattern is another strong clue for Bacteroids, then key biochemicals. Oxidase is negative, that's consistent for the whole genus. But importantly, B. stercoris is typically indole positive.
And that helps separate it from
Bacteroides fragilis itself, which is the most famous member of the group and is usually indole negative. So that indole test is a useful differentiator within the group. And finally, carbohydrate fermentation patterns. It typically ferments glucose, lactose, sucrose, rhamnose, and xylose. But it's negative for arabinose and salicin. These profiles can help pin down the species ID using biochemical test panels.
So these tests, like the potency discs, are actually direct windows into its natural built-in defenses, its intrinsic resistance.
Exactly. They're phenotypic readouts of underlying intrinsic resistance mechanisms. Take the aminoglycosides, like kanamycin or gentamicin. Bacteroids are universally resistant.
Why is that?
It's physiology. Aminoglycoside uptake into bacterial cells requires an oxygen-dependent electron transport chain to power the transport. Obligate anaerobes like B. stercoris simply don't have that system because they don't use oxygen, so the drug can't get inside effectively. No oxygen, no uptake, no killing. Simple as that.
Okay, that makes sense. What about the beta-lactomes, penicillins, cephalosporins?
Right. Almost all members of the B. fragilis group, including Stercoris, are intrinsically resistant to basic penicillins and many early-generation cephalosporins. This is due to that chromosomally encoded class A cephalosporinase. We mentioned the CIMP gene product. It's robust, often inducible, and very effective at breaking down those antibiotics.
But there's a key vulnerability there, isn't there?
Yes, critically important for therapy. That SEPA enzyme is inhibited by the common beta-lactamase inhibitors, clavulanate, sulbactam, tazobactam.
Which is why drugs like augmentin, amoxicillin clavulanate, or zosyn, piperacillin tazobactam, work against them.
Precisely. The inhibitor protects the partner beta-lactam from being destroyed by the CYP enzyme, allowing it to kill the bacterium. It's a perfect example of combination therapy overcoming intrinsic resistance.
Okay, that covers the built-in resistance. But this is where things get really complex, isn't it? Because Bacteroides stercoris isn't just intrinsically resistant. It's a master, apparently, of acquiring new defenses. This feels like the heart of the therapeutic challenge.
It really is. Its ability to acquire resistance genes is formidable, and it happens mostly through horizontal gene transfer, picking up mobile genetic elements from other bacteria in that crowded gut environment.
Let's talk specifics. Carbapenems are huge guns in infectious disease. How does resistance happen there? I know it's rarer in Stercoris than Fragilis, but still a concern.
It is a major concern, even if less frequent. When B. stercoris is carbapenem resistant, it's almost always due to acquiring a gene called CAIA, sometimes called CFIA. This gene encodes a class B metallo-beta-lactamase, or MBL. These MBLs are powerful enzymes that can chew up carbapenems. But here's the really interesting and slightly terrifying part about CFIA. The gene itself, as it sits on the chromosome or plasmid, often lacks a working promoter sequence right upstream of it.
Meaning it's there, but not really turned on?
Exactly. It's present, but not expressed at high levels. To get high-level expression and actual clinical resistance, a mobile insertion sequence, an IS element, needs to jump in. These IS elements often carry their own strong promoters. If an IS element lands in just the right spot, in the right orientation, upstream of CAFA, its promoter will drive high expression of the MBL, and boom, you get resistance.
Wow. So you could have a situation where
the lab detects the CAIA gene by PCR, maybe. So the potential for resistance is there. But if you do phenotypic testing, like an MIC, the bug might actually test susceptible to carbapenems because that IS element isn't in place yet.
That sounds like a ticking time bomb.
It kind of is. It raises the scary possibility that if you treat such a patient with a carbapenem, the antibiotic pressure itself could select for bacteria where that insertion event has happened, leading to resistance emerging during therapy, treatment failure. It's a huge diagnostic and therapeutic challenge, potential versus expressed resistance.
That's incredibly sneaky. What about metronidazole? That's such a workhorse for anaerobic infections. Is resistance common?
Thankfully, high-level metronidazole resistance is still uncommon in B. stercoris. But it's critically important when it does occur, precisely because metronidazole is a cornerstone therapy. Resistance is mediated by acquiring NIM genes. There's a whole family, NIM-A through NIM-G. These genes encode a 5-nitramidazole reductase enzyme.
And that enzyme does what?
It inactivates metronidazole. Metronidazole needs to be chemically reduced inside the bacterial cell to form toxic radicals that damage DNA. The NIM enzyme basically prevents that activation step. And like CFIA, these NIM genes are often found on mobile genetic elements so they can spread. We have seen documented clinical cases of NIMD-positive, metronidazole-resistant B. stercoris, so it definitely happens in patients.
Okay, less common but very significant if present. Now, clindamycin. I hear about resistance to that one a lot with the B. fragilis group.
You do, and for good reason. Clindamycin resistance is now unfortunately quite common in B. stercoris and the group overall. Surveillance data often shows rates well over 50% in many places.
What's the mechanism there?
The main mechanism is target site modification. Clindamycin works by binding to the bacterial ribosome, specifically the 23S rRNA component of the large subunit, and shutting down protein synthesis. Resistance comes from acquiring ERM genes. Erythromycin ribosome methylase genes, ERM, F, and ERMG, are particularly common in bacteroids. These enzymes add a methyl group to the 23S rRNA.
And that methyl group physically blocks clindamycin from binding.
Exactly. It changes the shape of the binding site just enough so clindamycin can't latch on effectively. And again, these ERM genes are very often carried on mobile elements, plasmids, and transposons, which explains why resistance has spread so widely.
It's just relentless acquisition. What about other drug classes, tetracyclines, fluoroquinolones?
Tetracycline resistance is practically universal now in clinical bacteroids. Bacteroids isolates. The dominant mechanism is ribosomal protection, driven by the TETQ gene. This gene product basically shields the ribosome from tetracycline. And TETQ is famously located on these incredibly efficient conjugative transposons that spread like wildfire through bacterial populations. For fluoroquinolones like ciprofloxacin or levofloxacin, resistance usually involves mutations in the target enzymes, DNA gyrase. Specifically, the gyrase subunit is a common spot, or it can involve increased activity of efflux pumps, systems that actively pump the antibiotic back out of the bacterial cell.
It's an incredible arsenal of resistance mechanisms across so many different drug classes. Is there an underlying reason why Bacteroids and Stercoris within that seem so adept at this?
It really boils down to opportunity and pressure. The opportunity comes from living in the gut microbiome, this incredibly dense, diverse soup of bacteria, constantly exchanging genetic material. It's a massive reservoir of resistance genes. The pressure comes from antibiotic use. Every time antibiotics are used, especially broad-spectrum ones, it selects for any bacteria carrying resistance genes, allowing them to flourish while susceptible neighbors die off. And because bacteroids are so abundant and readily engage in horizontal gene transfer, they are perfectly positioned to both acquire resistance genes from others and potentially pass them along. They are truly a key node in the resistance network of the gut, that sentinel role again. So with this constantly shifting landscape of resistance, standardized antimicrobial susceptibility testing, AST, becomes absolutely non-negotiable. We need it to guide treatment for individual patients, and we need it for surveillance to track trends.
And the main body setting the standards, at least in Europe, is EUCAST, right? The European Committee on Antimicrobial Susceptibility Testing.
That's right. EUCAST provides very detailed, annually updated guidelines on exactly how to perform AST for different bacteria, including anaerobes, and how to interpret the results using clinical breakpoints.
So for anaerobes like B. stercoris, what method does EUCAST recommend as the gold standard, and what are the critical conditions?
The reference method recommended by EUCAST for anaerobes is agar dilution. This involves preparing agar plates containing serial dilutions of the antibiotic. It's quite labor-intensive, though. It's performed using a specific medium called fastidious anaerobe agar, or FAA, supplemented with 5% horse blood. A validated alternative, much more common in routine labs, is disc diffusion using the same FAA medium. The absolute key, though, is incubation. It must be under strict anaerobic conditions at 35-37 degrees C. And the incubation times are specific, usually 48 hours for reading the MIC in agar dilution, or 18 plus or minus 2 hours for measuring zone sizes in disc diffusion. Getting these conditions right is paramount for reliable results.
Okay, you get your MIC value or your zone size. Then you need to interpret it. What are the EUCAST breakpoints? Let's use the latest, V14.0 from 2024, that we, as microbiologists, need to apply for bacteroid species like Stercoris.
Right. The breakpoints tell you if the isolate is susceptible, S, susceptible with increased exposure, I, or resistant, R.
That I category. What exactly does that mean clinically?
I means that clinical success is likely if drug exposure at the site of infection is increased. This could be achieved by using a higher dose of the antibiotic, maybe more frequent dosing, or if the drug concentrates well at the infection site. It guides the clinician to potentially optimize the dosing strategy for that bug. Now, something really important you'll notice in the EUCAST tables for bacteroids is the use of breakpoints and brackets for some drugs. Clindamycin and ertapenem are key examples.
What do the brackets signify?
Bracketed breakpoints are based on epidemiological cutoff values, or ECOFs, not primarily on clinical outcome data. An ECOF is the MIC value that statistically separates the wild-type population, bacteria without acquired resistance, from the non-wild-type population, those that have acquired resistance. ECOFs are incredibly valuable for surveillance, for tracking the emergence of resistance, but reporting an isolate as susceptible based on a bracketed breakpoint comes with a significant caveat. The clinical efficacy, especially if used as monotherapy, might be uncertain. There isn't strong clinical data correlating that MIC value with successful treatment outcomes.
That's a crucial distinction for interpreting reports. So let's run through some key breakpoints from UCASD 14.0 for bacteroids. Piperacillin tazobactam.
Okay, for Piptazo, MICS, EKM boundary L, R2 mg/L, zone diameter S, 24 millimeter, R24 millimeter, using a 306 RM disc.
Meropenem.
MICSR1, R1 mg/L, zone S is 29 millimeter, 10 gram disc.
Imipenem.
Very similar. MICSA1, R1 mg/L, zone S, 29 millimeters, 10 billion disc.
Ertapenem, the one often in brackets.
Ertapenem. MICSAir2, R2 mg/L, zone SE, R23 millimeter, 10 milligram disc. Note those brackets, EFF based.
Metronidazole, the workhorse.
Metronidazole, MICSA, 4 R4 milligl, zone SO, 25 millimeter, R25 millimeter, 5 millimeter disc. Resistance still rare, breakpoint is relatively high.
Clindamycin, also bracketed, right?
Yes, and with that clinical uncertainty caveat, clindamycin, MICSES air, R4 ml, zone SO pen, R10 millimeter, 2 nickel disc. Remember the potentially poor clinical correlation here.
And finally, amoxicillin clavulanic acid.
Amoxiclav, MICSA, R2 mg/L, zone SO 14 millimeter, R14 millimeters using a 21 Rodome disc. Knowing these specific numbers and especially understanding the meaning of the I category and those bracketed breakpoints is absolutely essential for accurate reporting and providing clinically relevant information.
Okay, let's bring this back from the lab to the patient. How are these complicated intra-abdominal infections, where B. stercoris is often involved, actually managed? Let's start with the Dutch perspective, the swab guidelines.
Right. Now, the source material didn't point to a specific swab guideline just for B. stercoris, but we can definitely pull together the approach for CIAI based on their general principles and guidelines for severe infections. The core philosophy of SWAB, the Dutch Working Party on Antibiotic Policy, is always about evidence-based therapy. That means using local resistance data whenever possible, and always aiming to de-escalate antibiotic spectrum once you know what you're dealing with. Stewardship is key.
What are the main principles that apply here?
Okay, several key things stand out. First, microbiological diagnosis is paramount. Get cultures from the infection site, blood cultures too, before starting antibiotics, if possible. This lets you target therapy later. Second, empiric therapy. You start broad, based on local guidelines and the likely suspects. For CIAI, that must cover gut flora, Enterobacterales, strep, Enterococci, and definitely the anaerobes like B. stercoris. Third, de-escalation. As soon as you get culture results and susceptibilities back, narrow the antibiotic coverage. Switch from the broad empiric shotgun to a targeted rifle. And fourth, IV to oral switch. Once the patient's clinically stable, usually after 48-72 hours, and they can take oral meds, switch them off 5E antibiotics. Reduces costs, risks, length of stay.
Makes sense. So based on those principles in typical Dutch practice, what empiric 5V regimens would likely be used for CII? Can you give specifics on drugs and doses?
Yeah, likely choices in the Netherlands would include amoxicillin clavulanic acid, a very common broad-spectrum choice. Adult 5E dose, typically 1,000 milligram amoxicillin, 200 milligram clavulanate every 8 hours. Another strong option is a second or third generation cephalosporin plus metronidazole. This gives good gram-negative coverage plus dedicated anaerobic coverage. So maybe Ceroxam 1,500 milligram of EQ 8 hours or ceftriaxone 2G IVQ 24 plus metronidazole 500 milligram IVQ 8 hour. And for more severe cases or maybe hospital-acquired infections where you worry about more resistant bugs like Pseudomonas, then Piperacillin tazobactam would likely be used.
And duration, how long do they typically treat?
SWAB guidelines, like most international ones now, emphasize short duration. For most CII with good source control, 4 to 7 days is usually enough. Longer courses generally don't help and just drive resistance. Now, zooming out to the international stage, the IDSA guidelines, Infectious Diseases Society of America, are hugely influential, often developed with surgical groups. They really emphasize a risk-stratified approach.
Risk stratified based on?
Based on how sick the patient is, things like their Apache 2 score. Are they in shock? Also their age, other health problems, comorbidities, and importantly, whether the infection started in the community or in a healthcare setting. Low risk versus high risk guides the initial therapy choices.
Okay, walk us through those international recommendations. Start with community-acquired CIAI, maybe mild to moderate severity.
Right. For community-acquired CIAI, mild to moderate, IDSA suggests several options. Single-agent regimens could be ertapenem, moxifloxacins, tigecycline, or tigeracillin clavulanate. Combination regimens usually involve metronidazole paired with something like cefazolin, cefoxam, ceftriaxone, or fluoroquinolone, ciprofloxacin, or levofloxacin. Now, here's a really crucial point reflecting resistance trends. IDSA explicitly advises against empirically using ampicillin sulbactam anymore because E. coli resistance is too high. And similarly, they advise against empiric cefotetin and clindamycin because of the rising resistance rates in the B. fragilis group itself. This shows how surveillance directly changes guidelines over time. Drugs fall out of favor as bugs evolve.
That's a critical update. What about the sicker patients? High risk or severe community-acquired CIAI?
For community-acquired CIAI, high risk severe, you need broader coverage, definitely including reliable activity against Pseudomonas aeruginosa and robust anaerobic coverage. So recommendations lean towards the carbapenems, imipenem cilastatin, meropenem doripenem, or piperacillin tazobactam. Alternatively, you could use a combination of an anti-pseudomonal cephalosporin like cefepime or ceftazidime plus metronidazole.
And healthcare-associated infections, always trickier.
Yeah, for healthcare-associated CIAI, empiric therapy must be guided by the local hospital's antibiogram because the risk of multi-drug-resistant organisms, MDRs, is much higher. You often start very broad, maybe a carbapenem or piptazo. And depending on the patient and local bugs, you might need to add coverage for MRSA like vancomycin or VRE like lanzolid or daptomycin.
And duration internationally, same emphasis on short courses.
Exactly the same. For most cases with adequate source control, 4 to 7 days of therapy is sufficient. Longer courses don't improve outcomes and just breed resistance. That message is pretty consistent globally now.
Okay, that covers adults. But children aren't just small adults, are they? Treatment needs careful tailoring, especially dosing. What are the key pediatric considerations for CIAI, potentially involving B. stercoris?
You're absolutely right. Pediatric treatment requires meticulous attention to weight-based dosing for both safety and efficacy. Plus, you need to choose agents with well-established safety records in children. Guidelines like those from Perth Children's Hospital, PCH, or pediatric IDSA sections provide specific advice.
So for a child, say, older than a month, with something like a perforated appendix or peritonitis, what are the empiric IV options?
It often depends on penicillin allergy status. For a child with no or low-risk penicillin allergy, standard options are IV amoxicillin clavulanic acid, dosed at 25 millikilograms of the amoxicillin component Q8, max 1G dose, or IV piperacillin tazobactam, 100 millikilograms of the piperacillin component Q8 ascorg dose. If there's a low-risk penicillin allergy, for instance, just a rash, a common regimen is IV ceftriaxone, 50 millikilograms Q24 hour, max 2G dose, plus IV metronidazole, 12.5 millikilograms Q12 hour, max 500 milligram dose. And for a high-risk penicillin allergy like anaphylaxis, the typical recommendation involves IV gentamicin plus IV clindamycin. Gentamicin dosing is age-dependent, actually at 7.5 millikilograms Q24 hour for 10Y, 7 millikilograms for 10Y with max doses. Clindamycin is usually 50 millikilograms Q8 dose, max 600 milligauss.
What about very young infants, neonates, with conditions like necrotizing enterocolitis, NEC? That's a really serious gut infection.
Or sometimes IV meropenem is used as monotherapy because of its very broad spectrum. Dosing in neonates is highly specialized, based on gestational age, postnatal age, and weight.
And like adults, the goal is to switch to oral therapy when possible.
Yes, absolutely. Once the child is clinically improving, febrile, and can reliably take oral medications, switching to an appropriate oral agent, like oral amoxicillin clavulanate, is standard practice to complete the treatment course. Okay, let's talk infection prevention and control, IPC. For Bacteroides stercoris, the approach is a bit different than for, say, MRSA or C. diff, right? Because it's mainly endogenous.
Meaning it comes from the patient's own gut flora.
Exactly. So the main focus isn't so much on preventing patient-to-patient spread in the typical way. It's more about preventing the initial infection from happening in the first place and managing the antimicrobial resistance threat it poses within the healthcare environment.
So given that focus, what are the core pillars of prevention and control for an organism like B. stercoris? You mentioned 3.
Yes, 3 main pillars. Pillar 1 is prevention of surgical site infections, SSIs. This is arguably the most effective strategy because most B. stercoris infections follow surgery. This involves meticulous adherence to SSI prevention bundles. Key elements. Surgical antimicrobial prophylaxis, SAP, is critical for GI or pelvic surgeries. The antibiotic chosen must cover anaerobes like afoxatin or maybe a cephalosporin plus metronidazole. Timing is crucial. Give it IV within 60 minutes before the incision. And keep it short, usually just 1 dose, or stop within 24 hours post-op. Beyond SAP, it's about aseptic technique during surgery, meticulous surgical practice to minimize tissue damage and contamination, proper hand-skin prep, maintaining the sterile field, All the basics done perfectly.
Okay, pillar 1 prevents SSIs. What's pillar 2?
Pillar 2 is standard precautions and environmental control. Even though it's endogenous, cross-contamination via healthcare workers' hands or contaminated surfaces is possible, especially with fecal matter involved. So hand hygiene is non-negotiable. Soap and water or alcohol rub after contact with wounds, drains, fecal material. Use of personal protective equipment, PPE gloves, gowns when anticipating contact with potentially contaminated fluids or surfaces as per standard precautions. And routine environmental cleaning and disinfection of patient care areas, especially high-touch surfaces or items like commodes that might see fecal contamination, helps reduce the overall microbial load.
Makes sense. And the third pillar sounds like it relates to resistance.
It absolutely does. Pillar 3 is robust antimicrobial stewardship. This is critical because the patient's own gut can become a reservoir for multi-drug resistant bacteroids. We need judicious use of all antibiotics, especially broad-spectrum ones, to minimize the selective pressure that drives resistance. Stewardship programs are key here, promoting therapy based on local data, using rapid diagnostics, pushing for de-escalation as soon as possible. It's about protecting the effectiveness of our current antibiotics.
That ties back nicely to the resistance discussion. And what about safety for us in the lab handling these
Good point. Bacteroid species are classified as biosafety level 2 or risk group 2 organisms. That means all work with cultures or potentially infectious patient samples must be done in a containment level 2 lab, following standard microbiological safety practices to prevent accidental exposures like needle sticks, splashes, or generating aerosols. Even our commensals deserve careful handling. And, these IPC strategies are always evolving. We absolutely welcome feedback from you, the listeners. Your insights, local experiences, suggestions for improvement, please share them in the comments. Let's make this a living, collaborative effort.
This is crucial for everyone in the medical microbiology space, definitely. So as we bring this deep dive to a close, it's really striking how Bacteroides stercoris serves as this perfect microcosm of the complex relationship we have with our gut microbes. It's a vital partner, essential for health.
Absolutely. Its role as a commensal, breaking down complex carbs, producing those beneficial SCFAs, that's incredibly important. But as we've spent a lot of time discussing, when it gets out of place, it becomes a significant opportunistic pathogen, driving serious, often polymicrobial anaerobic infections.
And the biggest challenge it throws at us today isn't necessarily its inherent ability to cause disease.
No, arguably the bigger challenge is its role as this major reservoir and shuttle for antimicrobial resistance genes. Its ability to soak up and potentially share resistance mechanisms for almost every relevant antibiotic class, including carbapenems, really cements its status as a key sentinel organism. Watching B. stercoris resistance tells us a lot about the broader resistance crisis brewing in the gut.
What's truly fascinating here is just how layered the approach to managing it needs to be. It starts way upstream with prevention, good surgery, proper prophylaxis, environmental hygiene.
And then, if infection happens, it demands that integrated response. Rapid, accurate lab diagnosis, getting that specimen right, using tools like BBE and MALDI-TOF effectively, then aggressive source control from the surgeons. And finally, smart, guideline-directed empiric therapy that anticipates polymicrobial infection and resistance, followed by de-escalation based on results. If we connect this to the bigger picture, the constant evolution of resistance in B. stercoris and the whole B. fragilis group highlights why ongoing surveillance, both local and global, is so vital. That data feeds directly back into treatment guidelines, keeping them relevant.
It really underscores the need for that constant interplay between the clinic, the operating room, the lab, and strong antimicrobial stewardship programs.
Exactly. It takes that whole coordinated effort, clinical skills, surgical precision, diagnostic accuracy, and that unwavering commitment to using antibiotics wisely.
So, as you, our listeners, go back to your labs and clinics, maybe this deep dive will make you pause and think a bit differently the next time Bacteroides stercoris pops up on a report. It really makes you wonder, doesn't it? What other common, seemingly quiet commensals might be lurking in the shadows of the microbiome, silently acquiring resistance, just waiting for their moment to become the next major challenge?